Let's start with our introductions of our first speaker. We have Anuraag Bukkuri, who is a third-year PhD student in integrated mathematical oncology. What a great title of an area, focus area, integrated mathematical oncology. He is at the Moffitt Cancer Center and the University of South Florida. He's working with Joel Brown. And of course, Moffitt, as a lot of you know, as you can see in the chat, is one of our organizers. So really exciting to be able to feature research from Moffitt. He's also currently a guest researcher in Lund University in Sweden. That's where he is now. So good afternoon, Anuraag. It's nice to have you here. He was an undergraduate at Dartmouth and then Minnesota. Scared off by all that cold weather, so he moved to Florida, I guess. I don't know, except now he's in Sweden, so maybe that doesn't make sense. His undergraduate work focused on math and computer science and biology. Okay, Anuraag, welcome to Club EvMed, and we look forward to your presentation.
Thanks so much, Charlie. Let me go ahead and share my screen here. Alrighty, great. Hopefully you guys can see this. Okay, so yeah, thanks again for the introduction. As Charlie mentioned, my name is Anuraag Bukkuri. I'm a PhD candidate at the Moffitt Cancer Center, and I'm currently at Lund University in Sweden with all of the cold. And so, you know, again, thanks to all the organizers and you know, I've been attending these events for quite some time. Obviously, I don't have as impressive of a record as Charlie or Meredith with you know, 60 and 71 talks attended, but I've definitely enjoyed the ones that I did attend and the annual conference. So I'm really happy to speak to you guys today a bit about some of the work that I'm doing related to state structure populations and cancer therapeutic resistance. So I first just want to start out with what I think of the state of cancer research today. So this is a quote from one of my favorite authors, Edgar Allan Poe, from his short story, The Murders in the Rue Morgue. So this is a detective talking about the abilities of the police force when they weren't able to solve, you know, a strange murder that happened in their local town. And so he says, he impaired his vision by holding the object too close. He might see perhaps one or two points with unusual clearness, but in doing so necessarily lost sight of the matter as a whole. And so I think this really aptly characterizes a lot of the current approaches that cancer research are taking to trying to understand cancer. So we're really obsessed with, you know, our favorite gene, favorite protein, favorite pathway, and all of these really new fancy shiny toys and technologies that we have without really, you know, understanding the broader picture and trying to get a bigger sense of what cancer is and how we can treat it. So along those lines, I sort of want to reframe cancer from thinking about it as a disease of the genes to thinking about it as a disease of evolution. And I might personally be biased here, but I think a great way for us to understand evolution is through math, right? So what is mathematical modeling? So I want to start out by saying that we are all modelers, right? Whether we are mathematicians, whether we are computer scientists, experimental biologists, or physicians, at the end of the day, we're all modelers. We're trying to understand cancer by analyzing some small subset of that, whether that be you know, a set of patients, some cells, mice, or really just equations. And so I like thinking of mathematical modeling as really a rigorous formalization of our conceptual model of reality. So it's not a model of reality, it's a model of what we think of reality. And so that gives it really this unique power in which we can think of our own ideas, our own hypotheses about how the world works, rigorously create a model for that, and then look at the implications of that. And perhaps if those don't correspond to reality, we can go back and reframe our thoughts and then, you know, proceed in this iterative process. So what's the point? Why do mathematical modeling instead of just, you know, looking at patients or cells or mice? And I think a lot of times when people ask that question about mathematical modeling, oftentimes they think, you know, it's great for data fitting. We have a lot of data, we need to process it, we need to understand it, and mathematical modeling is a way to do that, or is a way to predict future trends. And although those are really important, I sort of want to stress these following points for why we do mathematical modeling. So even if we just create a mathematical model and we don't do anything with it, we don't simulate it, we don't analyze it, I think that just that exercise, that process of creating a mathematical model really helps us understand our own thinking. because a lot of times we come up with hypotheses and ideas that we don't really fully understand what we're saying. And so when we create a mathematical model, it makes all of our assumptions explicit. There's nowhere to hide. Everything is out in the open. And so from that, you really understand, at least implicitly, what you're assuming. And in that process, that also leads to a lot more questions than answers, which is really, to me, the hallmark of a good research question or method. And so with that out of the way, let me just talk about, a small framework and, to model eco-evolutionary dynamics before diving into the cancer stuff. So this framework is called the G-function framework. It's very closely related to the sort of adaptive dynamics framework that's used in the literature. And it really just has two components. You have your ecological or population dynamics, and you have your evolutionary or trait dynamics. So the population dynamics is just a product of your current population size and what's called a G-function or a fitness generating function. And this G-function just, captures the per capita growth rate as a function of some focal individual, some cell, some animal strategy, the strategies of all the interacting individuals in the population and their densities. And then their evolutionary dynamics is just derived from that, from Fisher's fundamental theorem, just as a product of evolvability and the selection gradient. And I'll get a little bit more into what those mean in a bit. And again, there's some assumptions underlying this as underlies all sort of modeling frameworks, and I'll be happy to discuss that later. So how exactly do cells evolve? A really nice way to see how cells or animals or anything are evolving is using what's called this adaptive landscape that you can see here. So this adaptive landscape plots the fitness of cells as a function of their trait value. So this trait value could be anything from the number of transporters to drug resistance or something like this. And so how cells evolve, or if you imagine a cell starting here at this yellow circle, it's going to try to climb this peak of this adaptive landscape to get to this local peak at A. And how it's going to do that is, one, based on the evolvability, which is the capacity for a cell to respond to natural selection, right? You can think of this as a step size. What step size is it taking to climb up this landscape? And the other is the selection gradient, or dg/dv, which is really the slope of this adaptive landscape. So steeper your landscape is, the higher your evolvability is. Generally, you're going to evolve faster. And it's important to note that these two are usually quasi-independent. So the evolvability does not depend on the slope of the adaptive landscape. And in earlier work, we sort of investigated theoretically the importance of evolvability in a wide range of evolutionary contexts. And broadly, we noticed evolvability is really a double-edged sword. So in stressful environments, the benefits of having a high evolvability outweigh the costs. But in more stable environments, the opposite's true. It leads to mutational meltdown. You really don't want to be, for example, mutating a lot when you're pretty happy in your environment. But that led us to sort of the next question, which is, what if cells could actually modulate their evolvability in response to environmental conditions? And this has been seen, throughout the biological world. For example, this perspective paper by Susan Rosenberg and her team was looking at stress-induced mutagenesis in bacteria. So it's well known that bacteria under stress will increase their rate of mutagenesis to help cope with this stressor. So is there any way that this could happen in cancer? And it turns out that there might be, and that might happen through what's called the polyaneuploid cancer cell state. So what is this? This polyaneuploid cancer cell state, or PACC state, is formed when aneuploid or 2N+ cells bypass mitosis or cytokinesis or both and undergo some whole genome doubling. So what happens, the main characteristics of cells in this state is that they exit the cell cycle after S phase, which pauses division and allows them to avoid DNA damage or apoptosis. So it sort of serves as a refuge from therapy. And you also have an increase in your genomic content via this polyploidization program. And that leads to a generation of greater heritable variation that can then be dispensed onto their aneuploid progeny upon depolyploidization. And the really crucial thing here, which is what makes them so deadly, is there's a sort of condition-dependent formation, which means that cells in the PACC state, you'll observe them at really low levels during baseline conditions. But when the population is under stress, these become incredibly common. They constitute the vast majority of the population. And so we think that these PACCs can form facultatively, and so they can be modulated up or down as a strategy contingent upon the cancer cell's condition. So now we have to model it. So how can we do that? Well, the first thing we have to note is the PACC state actually exists as a cell state in the life history of a cancer cell. So these are not different cell types, right? That's a really crucial aspect. They're just a different cell state. So, just like butterflies and caterpillars aren't different species. They're part of the same species. They're just at different life cycles, right? And the same thing with human populations. Obviously, the butterfly and the caterpillar are going to respond to their environment in different ways, and that really matters for the fitness of the overall species. And so we need to find or create a way in which we can, incorporate both of these and then come up with, a nice model for that. And so to do that, we need to create a unifying fitness function to track the evolutional traits across, in our context, both these 2N or aneuploid cells and these PACC states. And this is a problem throughout the cancer literature. There are a lot of things that we treat implicitly as cell types that may actually be cell states. So it's something we should all keep in mind and think about carefully. But anyway, it turns out that the math that's needed to model, this sort of problem isn't really developed. And so we sort of have to develop the math to do that. And so how we do that, very briefly, is that we take all of our equations that we have, or maybe we have a life cycle graph like the people in life history theory like to do, and we encode all of that information into a matrix. And then we can use properties of matrix theory. Specifically, we look at the largest eigenvalue of this matrix, which really controls the long-term dynamics of the system, and we can use that then as our G function. And again, I won't go into the math because it can get really involved, especially when we look at things like convergent stability and if our state values are continuous, but again, happy to discuss that later. So with that modeling approach, we can then create a model first of the PACC ecology. I won't get into the equations here, but as you can see, these are sort of plug and play, right? A little bit like the click chemistry that we've been hearing about in the news recently, where we can basically just add on components or take away components really easily depending on the complexity that we want or what we really want to incorporate. So with that, we can then go ahead and simulate various hypotheses you might have on how PACCs contribute to therapeutic resistance. So one hypothesis is standard tumor heterogeneity. This is the standard hypothesis used in the cancer literature where there's no PACC state at all. We can look at, non-proliferation, which is sort of this hibernation strategy, or really evolutionary triage, which is sort of saying that the PACC state actually contributes to the evolution of resistance. And so in all of these plots, the top panel is going to capture the population dynamics or the ecological dynamics of the cells, and the bottom captures the evolution of resistance. So for standard tumor heterogeneity, basic thing we see is if you give a low dose of therapy, cells are just fine. You see a sharp drop in the population size initially, and then as the cells gain resistance, they increase their population size. When therapy is removed, they're all happy again. But when we give a high dose of therapy, they die. They can't evolve resistance fast enough. They go extinct. What we see with non-proliferation is that, when the therapy is administered, cells in the 2N state do crash, but a lot of them convert into this PACC state. So no evolution of resistance is happening in the state. It's just providing a refuge from therapy. And so we notice this sort of temporary state of resistance. Whereas with evolutionary triage, not only does the PACC state serve as a refuge from therapy, but it also, again, due to the polyploidization program, contributes to the evolvability, the evolution of resistance of these cells. So then what we see is that we see an increased therapeutic resistance strategy. And then as these cells are gaining resistance, the frequency of cells in the PACC state and the number of cells in the PACC state are gradually decreasing over time. So that's sort of what we see. We take the strong inference approach. We have all these hypotheses. Then we go into the lab and nothing actually matches, right? So what happens is, what happens in the lab is that under extreme stress, there is a dramatic transition of cells from the 2N state to the non-proliferative PACC state. But after a delay, cells in the PACC state begin depolyploidizing into progeny, but those progeny immediately display high levels of resistance to the stressor. So unlike we would expect under evolutionary triage, classic Darwinian evolution with a Mendelian inheritance, we do not observe the death of mutant clones. So it appears to be that the evolutionary process does not require creative destruction. It appears to solve the therapeutic problem without the mortality of less successful variants relative to more successful ones. So then we go back to the drawing board and we come up with a new hypothesis. And we say, perhaps it's some sort of self-genetic modification, right? So maybe via reverse transcriptional inheritance, changes that a cell undergoes during its own lifetime, for example, within the PACC state, might actually be written back into its DNA and then passed on to its offspring. The issue is that existing evolutionary models can't account for this because this is sort of a Lamarckian inheritance, I should say, in nature. So we have to, again, the math doesn't exist. We have to create some way of thinking about this. So what we do is we create what we call this birth-death switching process. Obviously, I won't go through this entire algorithmic flowchart because you guys will fall asleep if you haven't fallen asleep already. But the really important thing is really this green part. So what's happening is that once cells are exposed to therapy, they transition to the PACC state. When they're in the PACC state, the PACC says, okay, if I produce a progeny, right, is that progeny going to be happy in the new environment? And if that's the case, it'll go ahead and produce that progeny. That progeny goes ahead and continues this standard evolutionary triage process. However, if that cell is not going to be happy, it's going to die. What the PACC will do is, if the mutation conferred to that trait is better than the one it has, it'll sort of adopt that trait, and evolution will sort of proceed behind the scenes. So it'll continue to try to solve this problem until it has a good enough solution, and then they'll pop out and continue proliferating, right? That's the main crux of this. So how does this actually look? We can go ahead and simulate again. This is a high dose of therapy and compare the evolutionary triage that we saw before with the self-genetic modification. And what we see in the evolutionary triage is that once the high dose therapy is given, a lot of times, the population is not going to evolve resistance fast enough to remain extant in the population, right? And so what we see with self-genetic modification instead is actually all the cells survive, all the populations survive, and that's because they immediately enter this PACC state, and we see the stable PACC state, right, for these almost more than 100 time steps, 150 time steps. It's within the stable PACC state. It's trying to solve the problem. Once it does solve the problem, we notice this jump in the evolutionary dynamics. Those cells are relatively happy. They can continue proliferating, and the evolutionary triage process commences, right? The issue now is then, okay, we ask, why wouldn't cells always do this? If self-genetic modification is this cool, this awesome, why aren't cells always doing this, right? It turns out that there's one important aspect that we didn't consider, and that's sort of this whole density dependence between these two hypotheses. Because when cells are in the PACC state, they're not dividing, right? They're just staying there. And so what we do is we sort of run these in silico competition experiments between cell populations that evolve via evolutionary triage and between cell populations that evolve via self-genetic modification. And what we see is there are really three main regimes. So the first regime is sort of the neutral regime, where evolutionary triage, self-genetic modification, based on the threshold that we set, operates the exact same way. And what we see, as you'd expect, not really much of a difference in the number of extinctions that happen. In the green phase, that's sort of a low dose of therapy. And what we see is self-genetic modification goes extinct a lot more often because the evolutionary triage cells have this density-dependent advantage. But once you enter the high dose of therapy in the yellow regime, what we notice is that evolutionary triage goes extinct a lot more often, because at this point, you're not really caring about beating your neighbor. You just want to survive in your environment. And that's what self-genetic modification really allows you to do. So it's sort of a summary. I just want to say genes are definitely important. They aren't everything. We sort of critically need ideas from interdisciplinary fields, especially ecology and evolution. And then again, the common quotes, nothing in biology makes sense except in the light of evolution. And the evolutionary play then takes place in the ecological theater. And I also want to say that assumptions are critical, and we can't always treat cell types and cell states as the same thing. So currently what we're doing is we're probing these theoretical results in the lab. We're actually in the lab, we're doing experiments trying to test these hypotheses, various aspects of self-genetic modification, sort of a PACC memory that I haven't discussed and things like that. We're also investigating other aspects of PACC biology with complementary methods, more data-driven, agent-based modeling approaches. And we're also extending the application of the state structure modeling approach we have to other sort of questions of cell types and cell states in neuroblastoma, other things in broader ecological contexts like dispersal morphs. And we're also working on the theoretical framework and coming up with more proofs and sort of delving into that theory more. And so with that, if this is interesting to you, you can go ahead and go to this sort of web app that we've created to play around with G functions a little bit. And obviously, I just want to thank my team. So Emma Hammerland, a very interdisciplinary team. Emma Hammerland is a geobiologist here at Lund. Joel Brown is a squirrel biologist that you guys have heard from probably way too much. Ken Paietta is a cancer physician, a great physician there. Bob Austin is a physicist, and Sarah Ahmed is a cancer ecologist, experimental biologist. And then again, I just want to thank my funding from the Crawford Lung Nonska and KVA, and then my NSF grant as well. So with that, I'd want to thank you all for your attention. I can't see you, but hopefully you guys are awake again for the question and answer section, which I'm happy to do.
Thank you, Anuraag. That was fantastic.
Thanks.
Thank you for a great presentation. Really exciting to see all this work that you've been doing and this interdisciplinary team. So thank you very much for sharing it. Looks like we have one hand raised. Is that Jason Somarelli there? Go ahead, Jason.
It is. Hi, Charlie. But I see Elise, I think, beat me to it. So I'll defer to Elise, even though she's waving me off. Never mind. Sorry, that was great. I've been thinking about this problem a lot in the context of different drugs and how they exert sort of pressure or stress differently. And I was just curious what your so an example would be like like doxorubicin, which is basically, I mean, I'm glossing over a lot of stuff, but basically just targeting proliferation, right? Just anything that grows fast, it'll obliterate. Very cytotoxic drug versus something that's a targeted therapy like enzalutamide, a prostate cancer hormone therapy that's very cytostatic in nature, right? Its mechanism of tumor inhibition is very different. And so do you have a way to sort of address this in your model? What sorts of things are you able to do modeling wise to kind of capture the many different ways that drugs act and how they might differentially target or force populations into one of these states?
Yeah, so there's a couple aspects to that. So one aspect is sort of the eco-evo informed therapy aspect of it is sort of how we can combine these therapies in effective ways. And Fred and I have written a couple things sort of in our cancer corruption paper, another paper that we're currently working on now, and how we can sort of evolutionary trap these cells in various ways depending on their biomarker. And I've written another one on, for example, glucose uptakes from a more consumer resource sort of point of view. So there's definitely things we can do to sort of use these therapies in different ways. And also sort of, it's important to understand the sort of effects that targeted therapy and chemotherapy have in different ways. So for example, in our biomarker game, we sort of note that a lot of these biomarkers have functional roles, and chemotherapy is a lot more broad spectrum. So obviously that flattens out your adaptive landscape a lot more than targeted therapy, but it's more effective for a broader range of therapies. And so we hypothesize that perhaps you can give the chemotherapy, ramp up the biomarker production for these things, because a lot of those contribute to resistance to chemotherapy, and then hit it with targeted therapy and do that in a smart way. With regards to looking at this in the context of PACCs, it's something we're definitely doing right now. For example, even with PACC memory. So some of the experiments we're running now is that we think that cells that have entered the PACC state due to one therapy, right, the next time they're exposed to therapy, a completely different therapy, they could actually remember this PACC state and enter it more quickly. And so we're looking at that specifically in the context of like cisplatin and radiation therapy, and trying to understand, okay, how do these things differ? Definitely biologically, we like to assume that basically everything can induce PACCs, whether that's nutrient deprivation, hypoxia, radiation, chemo, whatever. But they definitely have a slightly different phenotypes. But that biology, we're really nailing down, whether that means the morphology of these cells, how motile they are, how spiky, and their morphology is different, and all of that. So that stuff biologically we're nailing down as well.
Thank you.
Yeah.
Okay, we have a question from Neil Greenspan. Neil, do you want to unmute and ask your question? Thanks for joining today.
Yeah, sure. So there are a number of other genetic or so-called epigenetic phenomena that cancer cells are known to exhibit, especially when faced with either chemotherapy or immunotherapy. And as I put in the chat, one would be metabolic quiescence, whereas genetic evolution requires a population, a single cell can change its transcription pattern and become metabolically less active in a way that subverts the effect of the chemotherapeutic agent, and/or increase expression of genes that encode molecules, for instance, that export the drug from the cell as it comes in. And then there are specific loci that get deleted, like in the case of HLA, in certain kinds of leukemias, where when you do some sort of immunotherapy, you can get loss of HLA, or just in response to the immune response of a patient. And then there is the concept of chromothripsis, which is sort of analogous to what you're talking about with aneuploidy, but it's essentially, I think it's a Greek word that means chromosome scrambling, which is a bizarre phenomenon. Exactly how that gets triggered is, I don't think, clear yet. But my point is that there's an enormous heterogeneity in the kinds of phenomena that can occur. And I think it's pretty tough to model a cancer in general, as opposed to particular tumors that have well-known patterns. I mean, I think your explanation of how models should be used and what they are was excellent. But, you know, I think one problem with modeling is trying to make the model realistic enough that it has a chance of sort of climbing its own evolutionary hill to a local peak.
Yeah, no, that's fair enough. And yeah, definitely there's sort of a trade-off between how general we want to be and how practical or useful it is in a specific circumstance. With regards to your more specific points, I would say, yeah, so PACCs are actually sort of a mechanism of plasticity, so it doesn't necessarily require genetic evolution that way, but definitely the other aspects that you considered, if we wanted to create more detailed models of that, we definitely could, and we just create more strategies that we would evolve. And so, yeah, definitely the way that we've modeled it, we've left that abstract. But generally, I mean, depending, however you evolve resistance, it's still sort of the same sort of peak climbing thing. So it is a qualitative model. We're definitely not claiming that we should go and apply this to a patient on the second floor of Moffitt immediately. There are definitely models that do that, but that's not the models we are. So we're just sort of trying to gain some qualitative understanding, make predictions, and then go biologically test them and sort of do that. But definitely your point is well taken, that there are a bunch of mechanisms that we haven't considered and that models can't consider that biology can.
Thanks.
So again, that's more the interplay between biology and math that really needs to happen, right? We can't do one without the other.
Okay, thanks, Neil. Thanks, Jason, for those questions. And Anuraag, thank you so much for a great presentation. We may have time at the end for a question or two additional, but I think for now we need to move on. And so let's thank Anuraag for a really great talk. That was fantastic. And I will introduce our second speaker in this showcase. It's Elise Paietta. She's a third-year PhD student in biology here at Duke University. She's in Ann Yoder's lab, and I've gotten to know her, at Duke over the past couple of years. It's really great to have her in our community here in evolutionary biology and ecology. She came to Duke with an undergraduate degree from Notre Dame. She worked in particular when she was at Notre Dame with the Amboseli Research, Baboon Research Project, and has done some great work there. And she's going to talk about her new work that she's doing since she's come to Duke. And part of this is through a TRISEM fellowship. And so we're through a research grant. So we're really excited about that as well. So go ahead, Elise. Thanks for joining.
Thanks so much, Charlie. All right, so yeah, my name is Elise Paietta. I'm a third-year PhD student in Duke Biology. I'm really interested in virus discovery and in transmission between humans and non-human primates. And I'm going to be talking about identifying diverse viruses using metagenomics. So why viruses? I think it's pretty evident from the pandemic that we've been living through, but viruses have global impacts. The majority of recent pandemics have all been due to virus transmission from animals to humans. Viruses have rapid evolution and an array of transmission modes and replication strategies that make them a big threat. And we've characterized less than 10% of viruses on the planet, leaving an over 90% knowledge gap in our understanding of viral diversity. And so why are we looking at viruses in lemurs? So lemurs are primates, so they have a close phylogenetic relationship with humans. There are over 100 species of lemurs that have evolved to take advantage of really different environments throughout Madagascar. And because lemurs are so diverse, we think that there are going to be particularly diverse viruses in lemurs also. There are also heightened human-lemur interactions in Madagascar because of conservation threats such as deforestation and damage from increasing magnitude of cyclones. And so these heightened human-lemur interactions create opportunities for disease transmission, including zoonotic disease transmission from lemurs to humans or from wildlife to humans, and then also zooanthropogenic pathogen transmission from humans to wildlife. And lemurs also have rapidly declining populations. So there are over 100 species of lemurs, about 98% of them are threatened with extinction. And only a handful of lemur-associated complete viral genomes are available. So we don't really know much about viruses in lemurs. So why is there this knowledge gap? So viruses can be pretty difficult to study. They're super diverse. There are no universally conserved sequences across viruses. And samples, there's usually a low quantity of viral nucleic acids. And a lot of viral nucleic acids, especially for RNA viruses, are pretty unstable. And so how do we overcome some of these difficulties? We can use viral metagenomics. So viral metagenomics is a method for virus discovery that's made possible by advancements in next generation sequencing and bioinformatics. But people really hadn't used viral metagenomics on multiple sample types for lemurs. And so we undertook a pilot study at the Duke Lemur Center in Durham, and we used a One Health approach. So we got blood, fecal, and saliva samples from multiple species of lemurs. We got whole mosquitoes from mosquitoes near the enclosures of the lemurs. We also got pest mice that are caught as part of the Lemur Center's pest protocols. We got saliva and fecal samples from them. And we also got saliva samples from the DLC staff members that worked closely with the lemurs that we were sampling. So after we got these samples, we extracted the viral nucleic acids, and for metagenomics, you separate the RNA viruses and the DNA viruses, extract them separately. And then we sent our extracted viral nucleic acids to the Duke Genomics Core for sequencing. And then we received those raw reads, and we trimmed and assembled the reads with Trim-O-Matic and Megahit. And then we used Diamond Blast to identify potential viral hits. And then we were able to annotate these virus genomes using SNOTE-TAKER2 and Vibrant. And then we refined our genome annotations with BLAST-N and BLAST-X. And then from there, we could construct phylogenetic trees to see what we were working with. And we found hundreds of viruses in the samples. And so I'm going to go over how we work with viruses from both a really well-known virus family and also how we work with viruses from lesser known families. So first, we found some papillomaviruses. So papillomaviruses are species and tissue-specific viruses. They are primarily found in mucosal and epithelial samples. They infect a diverse array of vertebrate hosts, including humans and non-human primates. They have varying pathogenic effects from invasive cancer to no effects at all. They have small circular double-stranded DNA genomes, about 6 to 8 kb. And PV species classification is based on L1 nucleotide sequence similarity. So L1 encodes for the major capsid protein of papillomaviruses, so that capsid of the virus. And it's the most conserved protein across all papillomaviruses. So that's why it's used for species classification, with 70% as the species demarcation threshold. And prior to this work, 0 complete genomes of papillomaviruses and lemurs were available. So we found 2 distinct papillomavirus species and 2 Varecia variegata, or black and white ruffed lemur, who's hanging here, individuals. So we found VAV-PV1, so Varecia variegata papillomavirus 1, and VAV-PV2. And so these two are more closely related to each other than any other known papillomavirus, but they still only have 63% L1 sequence identity. And so because that species demarcation threshold is 70%, we know that VAV-PV1 and VAV-PV2 are two distinct virus species. And VAV-PV1 was actually present in both lemurs. And so VAV-PV1 and lemur 1 versus 2 had 99.6% L1 sequence identity. And then one lemur had a co-infection with both VAV-PV1 and VAV-PV2. And if we're looking at the genome annotations, so L1 is that more conserved major capsid protein in navy blue. L2 in the lighter blue encodes for the minor capsid protein. E1 and E2 are regulatory proteins, and E6 and E7 are oncogenes. And so not only are the two papillomaviruses we found two new species, if we look at the phylogenetic tree, so we made a phylogenetic tree of concatenated amino acid sequences of E1, E2, and L1. And so you can see in the light blue are Varecia papillomaviruses, VAV-PV1 from that first individual, from that second individual, and then VAV-PV2. And so they form a putative new genus of the Papillomaviridae family, with the closest genus being the tau papillomavirus genus. And so from this, you can see how we can use viral metagenomics to discover new virus species and even a new genus within a pretty well-known virus family. And so we have deposited these viruses in GenBank and have hopefully a publication coming out, but we have characterized the first papillomaviruses in lemurs. And so next I'm going to talk about a lot less well-known virus phylum. So the Cresnaviruses. So Cresnaviruses are a newer virus phylum consisting of circular replication encoding single-stranded, which is the Cres DNA viruses. They infect pretty diverse hosts, including fungi, plants, animals, and diatoms. And all Cresnavirus genomes contain a replication-associated protein, the rep, and a capsid protein. So shown in green, rep is in red, and the rep is most conserved across all Cresnaviruses. So we pulled all the reps from our putative Cresnaviruses from our Duke Lemur Center dataset, along with all reps from known Cresnaviruses. And we used an enzyme similarity tool to generate sequence similarity networks for Cresnavirus reps and visualize this in Cytoscape. So what you're seeing here are all of these sequence similarity networks. So the most related rep proteins are placed in family level clusters. So each of these clusters is at the family level. So each node, so each colorful dot is a rep from the input. And then each edge, so each line is a measure of the similarity between two nodes. And our alignment score threshold was 60. So when it's greater than 60, there's an edge drawn between them. The red dots are the Cresnaviruses identified from my samples. And then these are some Cresnavirus known families that are better put together. And we're going to take a look at what we found in the Smackovirus family and Redondovirus family. So Smackoviruses are primarily found in the fecal samples of diverse vertebrates and invertebrates, but the actual host hasn't been confirmed yet. And only one potential host, a gut-associated archaeon, has been hypothesized through looking at CRISPR spacer sequences. And so when we look at the sequence similarity network, the known Smackoviruses are in pink, and the putative Smackoviruses found in one of the Duke Lemur Center samples is in red. And then potential Smackoviruses that haven't been confirmed are also in blue. Okay, and so we found really diverse Smackoviruses associated with multiple lemur species, including black and white ruffed lemurs, crowned lemurs, Shifaka, blue-eyed black lemurs, and collared lemurs. And so this shows that diverse Smackoviruses were associated with diverse lemur samples. So these were primarily found in lemur fecal samples, but there's also one in a saliva sample. And so that's just a look into how we work with identifying viruses from families that we don't know much about, and also just from one data set of around 50 samples, we were able to contribute a lot to what we know about smackovirus diversity. And we're also discovering novel viruses in human samples with metagenomics. So the redondoviruses are primarily found in the human respiratory tract. They remained undetected until recently due to their super divergent genomes. And one study found higher redondovirus levels in patients with periodontitis and critical illness. So if we look at this red dot right here, this is from a DLC staff saliva sample. And you can see that it's falling within the redondovirus family. And so, yeah, we're learning about viruses in human samples too with metagenomics. And so the outcomes of this pilot study. So with this pilot study, we successfully optimized our viral metagenomics methods for discovering really diverse viruses across many viral families in lemur, human, rodent, and arthropod samples, making our future work in Madagascar looking at viruses across the human-lemur interface possible. And we also characterized the first complete genomes of lemur papillomaviruses, and we contributed to our knowledge base of viruses in lemurs and humans, because remember, there's that huge knowledge gap in our understanding of viral diversity, so we're helping to contribute to that. And so for next steps, I am actually flying to Madagascar on Friday. So 4 days from now, I will be heading to Manumbu Special Reserve in southeastern Madagascar. This is a really unique forest consisting of lowland rainforest and littoral forest, where a lot of the plants and animals that live there only exist in that area. For example, this lemur, U. lemur cinericeps, only exists at Manumbu Special Reserve. And so this is really a hotspot of biodiversity. But Manumbu Special Reserve has undergone extensive deforestation and cyclone damage, creating an expanding human-lemur interface, thus creating opportunities for infectious disease transmission. And so I'm joining a really awesome, big interdisciplinary project that's looking at the interactions between human health, wildlife health, and forest health. And so I'll be working with Zoo New England and the NIH Premise Lab to do viral metagenomics on lemurs, rodents, domestic animals, and human samples. And we'll also be working with the non-governmental organization Health in Harmony. And Health in Harmony, what they do is they go into a community and they ask the community what they need in order to reduce logging in the area and reduce killing of wildlife. And so Health in Harmony is providing the communities around Manumbu Special Reserve with a mobile healthcare system, food security, and also forest guardian jobs. And in return, people reduce their logging. And so we're looking at how that might impact infectious disease transmission by sampling all of these animals. And we'll be working with the NIH Premise Lab, which will do all of the extractions and sequencing based on the methodologies that we optimized in the pilot work. So that will be a really awesome project that I am really lucky to be a part of and be traveling to Madagascar. And so with viral metagenomics, we're going to be able to look for both those zoonotic and zoanthroponic viruses. And so, yeah, we're really interested in what we'll see, and we'll probably find super diverse viruses. And so I want to acknowledge the Duke Lemur Center, which was awesome, all the staff members that helped me with all of the lemur sampling, TRISEM, which funded me, Duke Biology, and Sigma Xi, which also funded me, and a huge thanks to Dr. Arvind Varsani, whose lab I worked really closely with. He is a leader in viral metagenomics, and so he was the one who really helped me to optimize my methods and characterize some of these awesome viruses. And then also want to thank my advisor, Ann Yoder. And Yeah, thank you. And thank you to Club EvMed for accepting my nomination to be here.
Excellent. Thank you, Elise. That was a great presentation. Thank you so much. We have some questions that are in the chat. Would any of you, Neil or Cynthia or Jason, like to ask a question? It looks like Neil has his hand up. Go ahead.
We also have Cynthia has asked a question. Cynthia, if you want to go ahead.
Okay, thanks. That was a great presentation. Thank you. And good luck in the field.
Thank you.
I am puzzled by how you can tell which direction the transmission occurred, if it did occur.
So at this point, so for the viruses that I was talking about, those are pretty species specific. And so there was not that we didn't find transmission among the different populations, although we were looking for it. But really, in order to determine whether cross-species transmission was occurring, you would have to have, so the reason we're sampling in Madagascar multiple populations is because if cross-species viral transmission is occurring, it should be present in let's say if it was occurring between lemurs and humans, we can compare the viruses in the lemur sample versus the one in the human sample and use phylogenetics to determine if cross-species transmission is occurring.
Thanks.
Yeah.
And if I could just ask another kind of related question, Elise, have you thought about sampling the environment? And would you use the same kind of methods, or would you use a more targeted approach? I mean, do you think that you could use this kind of metagenomics approach to also understand the environmental sources of disease for the human?
I know it's possible for like water and wastewater, so we could potentially take samples from that too. But yeah, there's also with the Manumbu project, someone will be sampling mosquitoes, and then we're also collecting ectoparasites that are on all of the lemurs, and we're going to do viral metagenomics on the ectoparasites too. So
awesome. Okay, Neil, would you like to ask your question?
Sure. So I was curious, do you have any evidence that these coronaviruses actually infect lemur cells or primate cells as opposed to, members of the microbiota?
Depends on the coronavirus family. So for the smackoviruses that I was talking about, they are actually hypothesized to infect a gut-associated methanogenic archaea. And so yeah, it depends on the family, but still like a part of the virome of the lemurs. So for the smackoviruses, no, we don't think that they are actually infecting lemur cells.
And are those the only two gene products, the one you mentioned?
The capsid
For the coronaviruses? The replication-related protein and the capsid protein, is that it?
So again, it'll depend on the coronaviruses are pretty diverse, but all coronaviruses have the capsid protein and the replication associated protein. So that's like the common thread. And then a lot of them have other ORFs that we don't really, they have unknown functions at this point, but yeah.
Okay. Yeah, I thought there might be other gene products.
Yeah, those are just the two that are shared across all the coronaviruses that we're really looking for.
Thanks.
Yeah.
I think we can fit in one more question if that's okay, Becca. Okay. Jason, did you want to ask your question? Are you there?
I'm here. Yeah. I just was curious if you had plans on doing any correlation at some point for, and it may just be that there's either too many lemurs have virus or too many have hepatocellular carcinoma, but if you're planning on correlating those two at any point, or looking at the association for any enrichment for any virus in cancer.
So yeah, we really just looked for just papillomas on the viruses. So the vets at the Lemur Center did check for papillomas on the lemurs because, we knew which individuals we were sampling. And then when we go to the field, we'll also look for like clinical manifestations, but as of right now, nothing further than that. But yeah.
Yeah, I could see a really cool sort of correlative analysis later on down the road where you profile viruses, you don't know that they have cancer yet, and 2 years go by, and then you can start to make correlations between certain viral enrichment, sequence enrichment, and the onset of cancer as a sort of perspective thing. But it's a great tool set. Well done.
Thank you.
Okay, thanks for those great questions, Cynthia, Neil, Jason. Great to hear from you guys. Thank you, Elise, for a fantastic talk. Thank you again. Anuraag, really great to hear about your work as well. Thanks for joining us today. Again, I want to thank Meredith for all of her hard work on Club EvMed. For those of you who might have joined a little bit late, this is her last time helping us host this event. It's our 71st of these events, and she's been to every one of them. And Becca Cook, who's also on, will be taking over from Meredith. So thank you so much, Meredith, for everything you've done for Club EvMed and for Trisome.